Osmosis is the net movement of water from a higher to a lower water potential across a partially permeable membrane. Let's look at the term water potential first. Pure water would have the highest possible water potential. The more solutes that are dissolved in the water, the lower the water potential becomes. For example, a 5 mol per decimetre solution of glucose would have a lower water potential than a 2 mol per decimetre solution. If these two solutions were separated by a partially permeable membrane, the net movement of water would be from the less concentrated to the more concentrated glucose solution. Now let's have a look at how water potential gradients can affect animal cells. If a cell is placed into an isotonic solution, this means the water potential outside the cell is equal to the water potential inside of the cell. There would still be movement of water in and out of the cell across the cell surface membrane, but these would be equal, meaning that there's no net movement. If a cell is in a hypertonic solution, this means that there is a water potential gradient from the inside to the outside. Water moves out of the cell by osmosis, down the water potential gradient, and it shrivels up. If a cell is in a hypotonic solution, this means that there is a water potential gradient from the outside to the inside, so water moves into the cell by osmosis and it swells up and maybe even bursts. This is why it's really important that glucose is stored in an insoluble form like glycogen, because only water-soluble substances affect water potential. Follow me for more A-level biology content overviews coming soon.